Good afternoon. This is Kallaya Sritunyalucksana. I'm from Biotech Nasta, Thailand. Today, I will present to you the information of a new emerging shrimp viral disease called the decapod iridescent virus, or DIV1, under the topic of response to the threat of DIV1 in shrimp aquaculture. Let me first telling you the discovery of the DIV1. DIV1 is a formal name assigned by this ICTV. It was first detected in 2014 in the freshwater crayfish, quadriculinatus, by the team of Professor Fenley in Institute of Oceanography in China, and they named it the CQIV after the name of the animal, infected animal. Later in the same year, a team of Professor Huang Ji at Yellow Sea Fishery Research Institute reported a newly discovered iridescent virus that caused severe disease and high mortality in farm Pinus bonamai in another region of China, and they call it this shrimp hemocyte iridescent virus. So now the name of DIV1 was assigned by the ICTV with two isolates of CQIV and SHIV. The DIV1 has its virion as a typical icosahedral structure with a diameter of 150 nanometers. As you can see by the TM image in this slide, its genome is double-stranded DNA of 166 kilobase pair. Currently, known susceptible species including the oriental prawn, Exopalemon carinicauda, the Australian freshwater crayfish, quadriculinatus, the less swam crayfish, Procambalus kakii, the white leg shrimp, the Pinus bonamai, and also the prawn of genus Macrobacillium, including Macrobacillium nipponensis and Macrobacillium rosenbergii. There are also two species of crab have been reported to be experimentally infected with the DIV1 in the laboratory. The disease caused by DIV1 results in massive mortality accompanied by the growth sign of disease characterized by the hepatopancreatic atrophy with fading color. The infected animal usually has a soft shell with empty stomach and guts. The moribund shrimp sinks to the bottom, and the mortality in the pond can be up to 80%. The growth size of the DIV1 infection is not clear in the peanut shrimp, but in the Macrobacillium rosenbergii, you can see this in the figure here, the white area inside the carapace at the base of the rostrum, which will be clearer shown than in the peanut shrimp. So in the laboratory challenge, it revealed high mortality of Chilax quadriculinatus, the Procambalus kakii, and little Pinus bonamai injected with the DIV1 population. The mortality rate can be seen with 100% or zero survival within 14 to 20 days. On your right hand side, to determine the root of the DIV1 infection, the challenge routes here was including the immersion challenge, the reverse gavage, and also the per hour which are resulted in all 100% mortality in the penis vanami. The histopathology signs of the DIV1 infection include this pathognomonic lesion characterized by the presence of unique basophilic cytoplasmic inclusion, which shown here in white, along with this karyolectic and pycnotic nuclei, which shown in black. The target organ of the DIV1 infection is including the hematopoietic tissues, gills, hepatopancreas, periopods, lymphoid organ, and muscle. The nested PCR for the DIV1 infection has been developed using this ATPase primer, and the inventor used them to screen this farm animal. They collect 625 farm animals, including Lithopenes bonamai, Penis chinensis, Macrobacterium lucenbergii, and Macrobacterium japonicus. The Penis japonicus. And it found that within 625 samples tested, 99 of them were found positive, and with different positive rate. For example, as you can see in this table, the highest positive rate was found in the farm Macrobacterium lucenbergii of 50%, whereas the shrimp Penis bonamai and chinensis is about 15%. With the method that Q developed in 2017 based on this ATPase gene, we use them to screen our broodstock in a program to establish a breeding stock from wild capture specimen of P. monodon from the Indian Ocean. Our group participated by screening individuals from capture batch for a list of 40 known viral pathogens and parasites by non-destructive PCR methods, while they were being held in a biosecure facility. In April 2018, we detect this DIV1 in one batch. However, to our surprise, all of these ATPase positive samples were grossly normal and show no gross sign of DIV1 infection. Besides, the histology of this shrimp show normal hepatopancreatic tissue and the lymphoid organs. The surprise results come with the in situ hybridization. Usually, the positive reaction of the DIV1 using the DIV Dick label probe, showing the positive reaction occur in the connective tissue in the interstitial space between the hepatopancreatic tubules. However, in the wild broodstock with positive PCR for the DIV1, the positive reaction occur not in the cytoplasm but in the nuclei of the tubule matrix cells and cells in the spheroid. So from this work, we show you that the wild captures P. monodon broodstock from Indian Ocean are positive DIV1 by using this ATPase PCR detection. There is no gross sign of DIV1 or no histological lesion in this positive PCR broodstock. However, this curious in situ hybridization signal that found in the nuclei of infected cell is interesting. Is this suggests the pathogenic difference among these geographical regions? The more research has to be done. We have published this study in the preprint journal bioarchive along with the peer-reviewed journal, so you can look at this study in the bioarchive now. So, in conclusion, for the recommendation, so the surveillance program, the postdoc or post-surgery quarantine and health certification for ID1 are urgently needed in major shrimp-producing countries. And the emergence of the IV1 should lead to the warning that polyculture of different species of crustacean may bring high risk for high spread of disease, increase of susceptible species, and evolution of the pathogens. The group of Kui in 2018 also reported that the live polychaete is found positive for DIV1. So, the live feed should be checked or decontaminated prior to use. From our result, we show that the wild capture monodon from the Indian Ocean that intend to use as a postdoc in our program were found positive DIV1. So, to warn that this wild capture P. monodon should be subjected to PCR testing for DIV1 before use. However, we would recommend the domesticated stock for P. monodon SPF for DIV1 than the wild capture bull stock. So that's all my presentation. Thank you for your attention.